Welcome to Halteres Presents, a brand agnostic interview podcast that seeks to objectively highlight the happenings within the world of diagnostics. And now, your hosts, Rich Thayer and Mickey Urdea.
Welcome to Halteres Presents. This is Mickey Urdea for Halteres Associates. I'm the founding partner.
And this is Rich Thayer, the managing partner at Halteres.
Today we're interviewing Dr. Eric Klein, Distinguished Scientist at GRAIL. We had a wonderful conversation with him about so many aspects of MCED, or MCED as they say, which is multicancer early detection. So they've been pioneer in this space. They've had an opportunity to do a large number of trials, probably more than anyone else at this point, with nearly 400,000 patients. And they have learned a great deal, which Eric lays out beautifully for us.
And now our interview with Dr. Eric Klein.
Hello, Eric. Thank you so much for being on our podcast today. We're really quite honored to have you as a guest. Please tell our listeners a bit about yourself and your work at GRAIL.
Well, thanks for having me. I'm a urologic oncologist by training, and I spent 3.5 decades treating urologic cancer patients at the Cleveland Clinic. And in 2016, someone from this newly established company called GRAIL came knocking on my door, someone that I had a relationship with from another biomarker company, and said, I just joined this new company, and we're trying to do something really unique and new, which is to develop a blood test that screens for multiple different kinds of cancers. Would you like to help us? And I said, yes. And so I was part of the very first foundational study that GRAIL did called the Circulating Cell Genome Atlas, CCGA, which is a large case control study where the fundamentals of the technology were tested for the first time. And following that, while I was still practicing, I joined a second study, an implementation study called Pathfinder, where we looked at how the technology worked in the real world in ambulatory patients. And coincidentally, along the way, I decided to retire from medicine. I did a sabbatical at Stanford University, and I became a consultant for GRAIL, which was nearby Stanford. And at the end of that time, I was offered a job at GRAIL as a distinguished scientist to help further develop this technology and implement it and explain it to clinicians and patients and so forth. And so I've been with the company for about 2.5 years now.
Excellent. Thank you. So if you could help us understand from a technical point of view how GRAIL got to this current manifestation of the test, what kinds of markers and platforms and how's it evolved over time as you learn more?
Well, let's talk about GRAIL's origin story, which is really fascinating. In the mid-2010s, I think, or even a little earlier than that, Illumina, the company that makes all the big sequencing equipment that was used to sequence the human genome, was running a non-invasive prenatal testing service. And for the first time was looking for fetal trisomies in maternal blood. And that's what sequencing allowed the field to get to. And so there was a woman named Meredith Hawks Miller who was running this NIPT lab, who looked at, I want to say, around 125,000 individual samples of pregnant mothers looking for fetal trisomies. And out of that, she noticed that there were 10 that were outliers that had funny looking DNA in the maternal blood. It wasn't fetal trisomy, and it wasn't normal maternal DNA. And she was savvy enough to take this really minuscule signal to a very smart scientist and showed it to him. And he said, that looks like cancer DNA. You better go back and contact the obstetricians who referred these patients for blood testing and have them follow up with their patients. Remarkably, all 10 of these patients turned out to have cancer. What's remarkable is they didn't have any risk factors. They were all under 40, healthy women, asymptomatic, and they all had cancer, and they all had a cancer signal detected in their blood. And that was the origin story of GRAIL, which I think is really a remarkable story. So if you look at the broader picture, we know that cancer sheds all sorts of interesting biological signals into the blood. It's not only DNA, but it's RNA and proteins and exosomes and extracellular vesicles and all sorts of things. So that's been known for a long time. But based on the work that was done in the NIPT lab, GRAIL has focused on looking at what's called cell-free DNA, which is DNA that comes from both normal cells and cancer cells. And cancer cells shed them in the bloodstream as they develop aggressive biologic features and access into the bloodstream. And that's what the signal is that we're looking for. I want to emphasize that cell-free DNA isn't the only marker for cancer in the bloodstream, but it is the one that GRAIL has exploited in a very useful way. And the signal we look for in the DNA, it's really two things. One is, can we distinguish cancer DNA from normal DNA, just the same way the NIPT lab? And the answer is absolutely yes. We can do that using a signal called methylation. And then the second thing that's a unique feature, or was a unique feature of the test, being the first on the market, is the ability with more than 90% accuracy in someone who has cancer DNA in their bloodstream is to predict what organ system the cancer comes from. And so you think about this technology is like fingerprints. There are two fingerprints we can detect in the blood. One is a fingerprint of cancer versus normal, and then a fingerprint of, in the individual who has a cancer signal, what's the likely organ or organ of origin or anatomic origin or cancer type. And that's really powerful technology, and that is the basis of the test that has been used in our clinical development program, our research studies, and is available commercially.
What distinguishes a cancer methylation versus a non, how you differentiate, because of course normal DNA is also methylated.
Yes. So methylation is a fundamental biologic process for normal cells and cancer cells. It's also considered what's called a hallmark of cancer, so abnormal methylation patterns. So you have to go back to high school chemistry, I think, to understand this, which is to remember what a methyl group is, which was a carbon molecule attached to 3 hydrogens. So how do cells use that? Cells use that to attach methyl groups to genes to turn them on or turn them off. And a gross oversimplification, the way I think about it, and is easy to think about, is take a cell that wants to be a liver cell. It uses methylation to turn on all the genes rather, that make it a liver cell, and turn off all the genes that would make it something else. And a prostate cell does the same thing. It uses methylation to turn on all the genes that make it a prostate cell and turn off all the genes that would make it a liver cell. So you can think about it like multiple light switches on the wall, turning them on and off. Okay, so that's the normal biologic process. How does cancer use methylation? It's the same chemistry. It's attaching a methyl group to a promoter region on a gene. But what cancer does is to turn on all the genes that allow it to grow in an unrestrained manner. That's the hallmark of cancer. And it turns off all the genes that prevent it from doing that. And so that methylation pattern is different than the methylation pattern of a normal cell that doesn't turn on all the growth promoting genes and turn off all the growth inhibiting genes, but turns on different genes. So there are different patterns of methylation in cancer versus non-cancer. And then using the same methylation signal, you can, in a cancer cell, tell what kind of cell it's derived from. So you can tell a liver cancer cell from a prostate cancer cell, for example. And that is the basis of the AI algorithm. It's really 2 algorithms, and actually we've added a third recently, but the 2 basic algorithms, the core of the test are, is there cancer DNA present or not? We call that a cancer signal present. And then in those who have a cancer signal present, what signal is suggested as the cancer of origin, which is really useful clinically, because if you know where the cancer is coming from, it makes it easy to focus in on a diagnostic algorithm that says, if the signal is positive and it suggests kidney cancer, well, I better go look at the kidneys with a CT scan or an ultrasound to start with.
And so often that is the next thing done, right? Is some confirmation with an imaging method of one sort or another.
So all cancer screening tests, including the ones that we use currently as standard of care, are screening tests. They're not diagnoses. And so they raise the issue that this individual might have cancer if they have a positive screening test. And they all require a diagnostic evaluation to prove whether or not a cancer is present. So let's talk about mammography. You see a spot on a mammogram, all of those women who have a positive mammogram end up undergoing an additional diagnostic procedure. Sometimes that's ultrasound, sometimes it's MRI, sometimes it's biopsy, but it's a diagnostic procedure. MCED works the same way. If you have a cancer signal detected, it doesn't absolutely mean that you have cancer, but it means the suspicion is high and that you deserve to have a diagnostic evaluation to determine if you do have cancer.
Right. When you look at this, the difference between one tissue and another tissue of origin, how different is it from one person to another? Is there a large set and you find a subset of those for individual patients, or how does that work?
Yeah, so that's different biology that GRAIL hasn't looked at specifically, other than to say that, yes, there are individual differences in methylation patterns. So your methylated DNA doesn't look like my methylated DNA.
Right.
On the other hand, what we have discovered through our the number of patients that we have tested in our clinical trials is that there is a common pattern across individuals. So two individuals who have cancer are going to have abnormally methylated DNA that says it's cancer. It may not be exactly the same methylation pattern, but it's distinguishable from normal. The second really fascinating observation, which is really a key biologic insight, is that that methylation signal that says I'm cancer is shared by multiple different cancer types. And so you don't need to know what kind of cancer someone might have to screen their blood for the presence of a cancer signal. That was a real eye-opener.
The scope of the undertaking of this discovery process at GRAIL, including the scale of the studies, is really quite remarkable. If I recall correctly, there have been about 8 clinical studies and around 400,000 subjects. What has GRAIL learned that is permitted by all of this scale?
Yeah, it's actually 9 clinical trials and
Wow.
just a little under 400,000 patients committed to those trials. We could take the next hour discussing what we've learned, but I'll focus on the key trials we did and what we learned from each one.
Please.
First one I mentioned, the CCGA study, and that was a case control study. We took 10,000 people who were known to have cancer. And we knew they had cancer. We knew what kind of cancer they had. We knew what stage of cancer they had. And we tested this algorithm in them and compared it to 5,000 individuals who we thought were healthy controls, who were not known to have cancer. And what we learned in that study was, yes, this blood test can detect the presence of cancer when it's present. Secondly, we learned that it can accurately predict, now over 90%, what the cancer type is. And we've talked about that a little bit already. Thirdly, and really importantly, we set the algorithm so that we would not be telling a large number of people who didn't have cancer that they might have cancer. So that's called specificity. And the algorithm is set for very, very, very high specificity so that only 0.5% of individuals who have a Galleri test will have a false positive. What that means in real numbers is about 1 in 200 people. That's really important when you want to use a screening test across a population, because if we're testing millions of people, we can't be telling hundreds of thousands that they might have cancer when in fact they don't. And let me put it in perspective. Specificity also translates into the false positive rate, right? That's really important. So the false positive rate for Galleri is 0.5%. As I mentioned, the specificity is 99.5%. Let's compare that to a currently used standard of care screening test. And we haven't done that head to head in the same population, but the point is this: mammography is widely used in the population. And I mentioned these women have to have a diagnostic evaluation. The specificity of mammography is far lower than it is for an MCED test, and the false positive rate is far higher. So it turns out there's another metric called positive predictive value for a screening test, which is what is the likelihood that someone who has a positive screening test actually has cancer. So again, let's talk about mammography. Positive predictive value for mammography in the US for an average woman at average risk is 5%, which means 19 out of every 20 women who have a positive mammogram do not have cancer. Only 1 does. And yet all 20 have to undergo a diagnostic evaluation. So you can think about, if you're going to screen for multiple cancers across a larger population, why it's so important to minimize the number of false positives and limit the number of unnecessary diagnostic evaluations and limit the amount of anxiety that people get when they get a positive test. And everything about Galleri is designed to do that.
Yeah, we've stressed this so much in other diseases as well. It's a common problem.
Yes, it is.
Whole
Yeah.
body imaging clinics, my
Yeah.
goodness.
Yes, all of it.
A cornerstone of proper test design.
Yeah.
Yes, absolutely. Yes, correct.
We really appreciate the way you focused on that, because that's been quite rare. I mean, some of the people we've been hearing talk about these are talking about specificity of 90%.
Yes,
We're kind of scratching our heads, not really
Right.
understanding that.
right.
Yeah.
Well, that a specificity that low would result in many, many, many more false positives and many, many more unnecessary radiation exposure and many more unnecessary diagnostic evaluations. So we have
Right.
purposefully set that bar very high.
Right, rightfully so. I think people don't necessarily think about the mechanics of how you do this balancing of sensitivity and specificity.
Yes.
I mean, most of our listeners are accustomed to rock curves, and you have an opportunity to slide around that rock curve to balance sensitivity and specificity. Often people do that just towards sensitivity and give up the specificity,
Right.
rarely vice versa. Can you just talk a little about that thinking at GRAIL?
Yeah, the whole issue is a little bit complicated when it comes to MCED because
Yeah,
lowering specificity doesn't increase sensitivity all that much. It depends on the shape of the ROC curve. Here's the issue. The issue really relates to what problem in cancer screening are you trying to address? So stage 4 cancers, metastatic cancers, only account for, I think it's about 18%, somewhere around 14 or 18% of all new cancer diagnoses in the US. But they account for almost half of all deaths, right? So most cancers that are diagnosed are stage 1 or 2, and one might logically think, well, you want a test that finds more stage 1 or 2 cancers. And that's actually not the way we're thinking about it. Really, what we want is not to overdiagnose early stage low-grade cancers that don't need treatment. What we want is to reduce the incidence, the occurrence of the advanced stage cancers that kill people. And that's the way our trials are designed. We have a randomized trial in the UK called NHS Galleri, where the endpoint is reduction in presentation with late stage cancer. Really, what we want is a test that finds those late stage cancers before they're lethal. We do not want to overdetect low-grade early-stage prostate, kidney, thyroid cancer. We could easily do that by setting sensitivity lower and increasing the number of stage 1 cancers that are detected. But those aren't the cancers that we care about because either they're not lethal or they don't need to be treated. So one of the interesting things about the MCED technology and methylation is the answer to the question, what cancers does Galleri detect? It detects cancers that need to be treated. And what we have found is that low-grade, low-stage cancers like early-stage kidney cancer, early-stage thyroid cancer, low-grade prostate cancer do not shed cell-free DNA into the bloodstream because they're not biologically aggressive. The ability to shed is a sign of biologic aggressiveness and a sign that these cancers need to be treated. So if you have a detectable cancer, a cancer that's detectable by MCED or methylation-based or Galleri-based technology, we really believe that it's one that needs to be treated. If you think about what problem are we trying to address here with MCED screening, let's talk about the status quo. And the status quo standard of care screening, we screen for 5 cancers. So breast, colon, lung in the high-risk population, cervical, and prostate cancer. All of those screening tests have been shown to reduce mortality to those cancers. And despite that, we still lose 600,000 people a year due to cancer. And the reason is that only 14% of all cancers in the United States are found by current screening tests, only 14%. And when you account for lack of adherence, particularly for lung cancer screening, more than 80% of cancer deaths come from unscreened cancers. So that is the big unmet need that MCED technology and Galleri is focused on, is addressing the fact that despite the fact that we have good screening tests that work for a limited number of cancers, we still lose 600,000 people a year. And so to get back to some of the clinical studies that we did, in the second study we did called Pathfinder, we tested the validated test that came out of the case control study prospectively in 6,600 ambulatory primary care patients. and we returned the results to the patients. What we found was that using Galleri or the MCED technology in that population doubled the number of cancers that would have been detected by standard of care, that half of those cancers were at stage 1 or 2, and more than 75% of them did not have standard of care screening tests. So we discovered a lot of biologically significant cancers that need treatment in asymptomatic patients at early stage that would otherwise not have been caught by standard of care screening. And that's what the Pathfinder study taught us. And that is really the key observation that led us to offer the test on the market, because we see real value in achieving those things that I mentioned, increasing the overall cancer detection rate, finding cancers early, and finding the ones that need to be treated, and finding ones that are not being addressed by the current standard of care. And in the Pathfinder study, what we learned was that we could detect many, not all, but many of those cancers that we don't screen for now that people potentially could die from.
This must be a difficult thing to make people understand, because most people are so focused on get those early stage, get to stage 1, stage 2.
Yes.
It's contrary to what most people would believe.
Yes. So it's not that we don't want to find stage 1 or 2 cancers, but
Yeah.
we want to find the aggressive cancers that would otherwise kill people at earlier stages, whether it's stage 1, 2, or 3. I mean, I just did a deep literature review. There has been huge progress in the last 5 to 10 years in treating stage 3 cancers. And so what used to be thought as uniformly fatal is no longer the case. There have been huge gains in lung cancer, and I mean, you name the cancer, there's been substantial improvement there. So part of the issue is, again, the goal is to find the cancer earlier, not necessarily at stage 1. And finding it at stage 1 could lead to overdiagnosis, and we don't want to do that. So as you mentioned already, alluded to, it is a careful balance. And we're still accumulating data on this, and we will have a great deal more data that will be released in the near future that will illuminate these issues even more. So the next study that we did is called Pathfinder 2, which is very similar to the Pathfinder study, but much bigger. It's 35,000 individuals screened once and followed for 3 years. And the first look at that data is going to be presented at the European Society of Medical Oncology meeting in late October. And then we have the only randomized trial of MCED, which is called NHS Galleri, which was done in the UK. And that was 140,000 individuals, 70,000 got standard of care screening, 70,000 got standard of care screening plus Galleri, and they were tested once a year for 3 years. And the results of that trial will be available sometime in the first half of next year. So there's lots more to learn here. I will say what's interesting is if you focus on the test performance characteristics, so the sensitivity for finding cancer, the positive predictive value, the very low false positive rate, we have seen robust performance across all of the studies that we've done, which is unusual. Usually when you do a case control study where you know who has cancer and who doesn't, you get very, very robust performance. And that tends to fall off in subsequent studies when you go out in the real world. We've actually seen the opposite. And in fact, there have been a couple of reports, if you just focus on positive predictive value, again, the likelihood that someone who has a positive test, a cancer signal detection, actually has cancer. In our big studies, CCGA and Pathfinder, the positive predictive value was in the mid 40%. The Mayo Clinic reported in a primary care journal earlier this year that in their use of Galleri in their population in primary care, the positive predictive value was over 70%. And the Dana-Farber Cancer Institute also reported at ASCO this last year, the American Society of Clinical Oncology, that they found that the positive predictive value in Galleri of patients referred to them was also over 70%. We know in our studies that there's a healthy volunteer bias. So individuals who volunteer to participate in studies are more likely to have taken care of themselves, less likely to have cancer. You know, positive predictive value depends to some degree on the prevalence of the cancer. So if you have fewer cancers in the population, the positive predictive value is going to be lower. We also know that the people in the studies were very adherent to standard of care screening. So they were up to date on their PSAs and their mammograms and their colonoscopies and so forth. Again, that would tend to cull out cancers that MCED would detect in a population that wasn't quite so adherent. So what we think is happening here, and we've seen the same thing in our real world data, is the positive predictive value is higher than it is in our studies because the population, while still skewed a little bit to people who have the means to pay for the test, is not quite as healthy volunteer bias kind of thing that we see in the studies. So we think as you go out into the broader population where the prevalence of cancer is higher because there are more risk factors, because people are less adherent with screening, there's less homogeneity and social determinants of health and so forth, that the positive predictive value goes up. But it's really, those are real world observations. So it's very interesting.
As we've shown, or as we've known from many other disease situations, for instance, during the pandemic, and as the disease, SARS-CoV-2 moved through the population, and positive predictive values change, depending on what the prevalence is likely to be out there. How have you accounted for that in screening for early stage versus late stage cancer? How have you adjusted your algorithm, or do you adjust the algorithm as the patient progresses through the disease states?
No, we do not adjust the algorithm at all based on those characteristics. But you've touched on a really important point. The performance characteristics of any given MCED test, any screening test, are going to depend on the details of the population where it's used. And one of the challenges of comparing different MCED tests is that the populations that they've been tested in aren't exactly the same. The distribution of cancers in the population is different. There might be age differences. There are social determinants of health that one cannot account for. Adherence to screening is a challenge. And so the field, the academic part of the field, is struggling a little bit with how to do robust comparisons of tests. And that's why it's important, if you're thinking about that, to focus on what are the test features and what does each individual test offer as an advantage, as opposed to just the performance characteristics. And so we can talk about the architecture of Galleri and what the potential advantages are there.
Eric, we found this in the past too. It's so little understood, this topic here. One assay may not work through the entire disease course, or if it does, how do you incorporate up front,
Right.
that understanding? So yeah, let's unpack that a little bit further, please.
Okay. So let's start with the observation that if you're unlucky enough to get cancer, you can't pick what cancer you get. So even if you're adherent to colonoscopy recommendation, screening guidelines, and you get your colonoscopy regularly and you get your mammography regularly, you're far more likely to be diagnosed with a cancer that you are not screened for than one you are screened for. And that's true even if you participate in a big clinical trial. So there are a number of big randomized clinical trials that have screened for lung cancer and ovarian cancer. And the individuals in those trials were more likely to die of things other than lung or ovarian cancer than they were of those cancers. So that's the first thing. And that suggests that the best MCED test, the ideal MCED test, is going to be one that screens for multiple different kinds of cancers, because again, you can't predict what cancer you might get. So there are some companies that are focused on smaller panels of only reporting out a signal that's detected of 10 cancers, say. I'm drawing that out of the air. I don't think that that's as valuable as a test that's been validated to detect multiples of that, dozens of different kinds of cancer. So that's the first test architecture feature that Galleri has. Then the second issue is if you have a blood test that suggests you have cancer, you have to do a diagnostic workup. And so you have to figure out, well, how do I find the cancer? So one approach to that is to not have what's called a cancer signal origin call, a prediction of where the cancer is. And there are some tests on the market that do that, where the suggestion is that you should then go to a whole body CT scan, and that's the best way to find the cancer. We've taken a different approach, which is to include as part of the test report in anybody who has a cancer signal detected, a cancer signal origin prediction, which I've mentioned already in the current test is about 93% accurate. That helps simplify the diagnostic evaluation. So if the CSO, as we call it, cancer signal origin prediction, I used this example already, suggests kidney, you look at the kidneys, you do a CT of the kidney. Why do you need to do a CT scan of the chest if you're not certain the patient has cancer to begin with? Go where the money is, as the bank robber Willie Sutton said, right? You got to go where the money is. Or if the CSO suggests colon cancer, what are you going to do? You're going to do a colonoscopy. Let me go back for a minute here. So let's talk about the CSO. So the diagnostic evaluation that's triggered by a CSO is what we call the targeted diagnostic evaluation. You look in the most likely place first. So if it's a lung CSO, you look at the lungs, chest CT. If it's kidney, you look at the kidneys. If it's ovarian, you might start with a transvaginal ultrasound or a pelvic ultrasound or a CT, something like that. It's very logical and easy, actually. Let's take a hypothetical patient, though, who has colon cancer, and you don't know it. You don't know that the patient has colon cancer, but this person exists and is being screened. So there are a number of different options for colon cancer screening. Fecal occult blood test. If you have a positive fecal occult blood test, you get a colonoscopy. That's according to guidelines. If you have a positive fecal immunohistochemical test, a FIT test, you get a colonoscopy per guidelines. If you have a positive Cologuard, you get a colonoscopy. If you have a positive Galleri test with a CSO prediction of colon cancer, you get a colonoscopy. If you have a positive MCED test that doesn't have a CSO, you get a whole body CT, which doesn't make any sense at all. And the same would be true for a woman with a suspicion of breast cancer, right? If you have someone who's a suspicion of breast cancer, you image the breasts, right? If you have an MCED test that doesn't have a CSO, then you get a whole body CT. And, the challenge is whole body CT, I don't have data to prove this, but I think it's intuitive, as a whole body CT is probably not as good a screen for breast cancer as mammography is. It's probably not as good a screen as colonoscopy is for colon cancer. So that's why we think there's real value to the CSO. The other value to the CSO is it really minimizes radiation exposures. There was a publication in one of the JAMA journals earlier this year that looked at overuse of imaging in medicine in the United States, not related to MCED, but just in general, and predicted that 3 to 5% of all cancers in the United States in the near future could be due to radiation exposure from diagnostic imaging for all sorts of things. Mm-hmm. And so one can argue about how much radiation is too much and how much is safe and that sort of thing. But you can't argue with the idea that less radiation is better than more radiation. And so using the architectural feature of a CSO call, half of the CSO predictions that we make, and there are potential 18 CSO predictions, half of them do not require any radiation for the initial diagnostic evaluation. And so when you couple that with a high likelihood, a high positive predictive value, the number of unnecessary radiation exposure is really going to be minimized by using a test that has a clinical signal origin prediction.
Yeah.
This episode of Halteres Presents is brought to you by the UC San Diego Extended Studies In Vitro Diagnostics Product Development course. Are you looking to take your biotech career to the next level? The UC San Diego Extended Studies In Vitro Diagnostics Product Development course is a unique, live online program designed for managers, entrepreneurs, investors, and scientists. This team-taught course by award-winning industry experts covers everything there is to know about the requirements for moving an IVD product from concept to regulatory approval and market launch. Explore real-world product development cases to help you plan for success by covering topics such as setting product requirements and specifications to meet customer needs, critical path management, resource planning, principles of assay development, instrumentation, role and composition of the product development team, risk analysis, and IVD clinical and regulatory issues such as compliance and complaint handling. Ready to get started? Enroll today and use our exclusive discount code to get a 10% discount on your enrollment. Use code HALTERES10 at checkout. That's HALTERES10 for 10% off the price of your UC San Diego Extended Studies In Vitro Diagnostics Product Development course. Just check the link in the show notes for this episode and use code HALTERES10 to enroll today. Accelerate your in vitro diagnostics career with the UC San Diego Extended Studies program. Your future in biotech starts here.
This is a very interesting conversation. I'm thinking back to Keytruda. Now, Keytruda was fascinating as a therapeutic for cancer. What I found particularly interesting was they used it in a number of different individual cancers to prove that it worked.
Mhm.
Then they went to the FDA and said, why can't we roll these up and have a pan-cancer claim? And of course, they did that. Now, that's not typically how people do
Right.
diagnostics, but de facto, you're almost going to tear it apart and go backwards because you have to have the best screening test for each cancer to roll it up to have the best MCAD.
Right. Yeah. Listen, this is a new world. We have to change the way people think about this. This is brand new technology, and it cannot be evaluated the same way that we evaluate single cancer screening tests.
Yeah.
And we're in a molecular era now. There was just a publication in Nature Communications, I think, or Nature Medicine from Italy, where they did a randomized trial of targeted therapy, molecularly targeted therapy, regardless of histology, versus standard of care therapy for several hundred patients. And they showed that the targeted therapy approach worked better.
Hmm.
I mean, the response rates were higher, and the initial suggestion is that progression-free survival is higher and that sort of thing. And so we're trying to, along with the targeted therapy field, try and get people to think about not individual cancers, but there is a molecular signal of cancer, not a specific cancer, in the bloodstream, and you have to think about it differently. Just the way
Yeah.
the idea that you just mentioned with Keytruda and the idea around basket trials, where your therapy is determined by the molecular features of your cancer rather than its anatomic location. It's a new world, and it's hard to change paradigms. It is. People are set in their ways.
And you need huge trials to make
Yes.
these things happen,
Yes.
as you learned.
Yes, absolutely. And committed to, and we're the biggest, no one else is committed to 400,000, to almost 400,000 patients in clinical trials.
Yeah.
And so to get back to what's the value of that and what have we learned? We've learned of the robustness of the methylation platform, the targeted methylation platform that we're using across multiple individuals, multiple cancer types, and multiple populations of patients in different circumstances, right? Case control studies, ambulatory primary care practices, in Pathfinder 2, and in NHS Galleri, a very, very diverse population with respect to measures of deprivation and social determinants of health and that sort of thing. And we've seen over and over how robust the platform is and how well it works across different populations. Really important to have that information, to have confidence in it.
Yeah.
Great discussion, Eric. Thank you. Let's shift gears a little bit. In your opinion, what needs to happen for MCED tests to become more broadly adapted, perhaps even mainstays of medical practice, either now as LDTs or in the future once they become FDA approved?
There are lots of potential barriers out there, one of which is we are challenging the status quo. And having worked at an academic institution previously, academic institutions don't like to be challenged, right? They have their view of the world that they believe is correct. And so we have to get past several hurdles here. One is there's a small but vocal community suggesting that we shouldn't be screening for any cancers. And so anytime there's a big MCED study published by anybody that comes out, we hear from that community, and it just creates this noise, which is, from a cancer doctor standpoint, really disturbing, because we know that screening works for the standard of care, cancers that we have. We know that as a strategy, screening for cancer works if you have the right screening test. So that's one barrier. A second barrier is getting people to understand the technology and to recognize that MCED technology works differently than single cancer screening tests do, and it has to be evaluated differently. So you cannot look at the performance of MCED for a single cancer and understand how it might work in a population. The goal here is to detect cancer at a population. We want to screen big populations without knowing what cancers are in there. And ultimately, we hope to reduce mortality due to that cancer. But you have to look at it in the aggregate in a population. You can't look at the effect of MCED on prostate cancer per se, or breast cancer per se, or colon cancer per se, because that's not how the population works. Again, in a population, the individuals can't pick what cancers they get. All those cancers are out there. So we believe that the most appropriate way to evaluate this is in the aggregate is what is the cancer detection rate in a population with MCED across all cancers, not for an individual cancer. And what's the effect on reducing the incidence of late-stage cancers in the population at presentation, as opposed to focusing on single cancer. So that's another battle that we're fighting. On a practical level, having FDA approval will certainly help because many physician guideline-based organizations don't endorse things until they're FDA approved. So that will help. Many insurance companies will not look at new tests or consider even reimbursing for them unless they're FDA approved, or at least that's what they say. And really, I think the big issue is MCED is if it's not paid for, if it's not reimbursed, then no company will have the resources to continue going to be able to make big investments in clinical trials to make it go. So having reimbursement, I think, will be a key issue also. If we can get past the reimbursement issue, then we can work on convincing, the physician community, the patient community, the academic community, and so forth. Okay, this test is widely available now because it's reimbursed. These are what the advantages are based on the studies. And that's what it's going to take, all of those things. So let me give you one specific issue that's really important. Screening tests like PSA, colonoscopy, and mammography are not required to be paid for by Medicare, except by the fact that Congress enacted laws that said Medicare must pay for these, and the private insurers followed. So there's no law that says MCED testing has to be paid for, but there is an important bill in Congress that has huge bipartisan support that would allow, not require, that would allow Medicare to reimburse for MCED. And that brings me to the very last clinical trial that we're doing that's still accruing, which is called the REACH trial, which is actually a CMS approved and actually paid for a trial. They're paying for the MCED tests. 50,000 individuals across a diverse population in the US, multiple different sites, screened once a year for up to 3 years, and compared to standard of care screening. So Medicare has really indicated an interest in learning about how this technology works and really wants to understand how it will be utilized and apply and help their population. So in the short term, I think the biggest issue is if we can get the MCED bill passed, that will really help drive adoption.
Yeah, it's a very useful discussion on barriers. It occurs to me, is there one more, as we have been witnessing with the rise in other types of genetic testing? There appears to be a dearth of genetic counselors. What do you do? How do you advise the patient, treatment pathway? I mean, just what should be next for the primary care docs and the specialists? Is there a corollary here in MCED testing?
Many people think of it that way, but I don't think that that's a true comparison. Here's why. We're not testing, MCEDs don't test someone for a disposition to cancer. MCEDs are a screening test like mammography that says, what's the likelihood that I have cancer right at this moment, not 10 years from now, not do I have a genetic predisposition to cancer that I have. And so it's really understanding the genetic predisposition and the risks there and the behaviors that might prevent it and the regular diagnostic evaluations and screening studies that need to be done in someone, let's say, who's a BRCA carrier who needs to be monitored before they have cancer for breast cancer or ovarian cancer, that sort of thing. That's completely different. MCED is actually pretty simple, and we have to get people to think of it as a simple screening test, okay? I have a blood test currently, a complete blood count. Am I anemic or not? That's the question that answers, right? I have a blood test that measures kidney function. Do I have, chronic kidney disease or do I have normal kidney function? If I have an MCED test, is there a cancer signal present or not? It's binary. And then because of the cancer signal origin prediction that we have, the diagnostic evaluations are easy because the report only has a single cancer signal origin prediction. So positive test, positive MCED test, cancer signal detected, single cancer signal of origin, let's say it's kidney, I know to look at the kidneys. It's actually quite simple. And I think it's just when people consider, learning about a new paradigm like this, it's easy to get overwhelmed. and to think that you have to be a genetic counselor or you have to be a molecular biologist to understand this, and you don't really. All we want people to understand, primary care physicians, is this is very robust technology that looks for a DNA-based cancer signal in the bloodstream. But on a practical level, it's very simple to interpret. Cancer signal detected or not. If there's no cancer signal detected, it's critical that you advise your patients two things. One, continue with standard of care screening. And two is, it's not an absolute certainty that you don't have cancer, right? You can miss a breast cancer with a mammogram. So it's important, but the negative predictive value is very high also. So it's important for the physician to have a general discussion about the importance of cancer screening and then add in the fact that you can now do an MCED test. It's just like before Cologuard came along, a stool-based test, for example. People did fecal occult blood testing or FIT testing and so forth. And then Cologuard came along. So what happens is the primary care physicians start talking about the importance of colon cancer screening. And here are your options. You can have a fecal occult blood test, you have a FIT test, you have a Cologuard test. Now there's blood-based testing for colorectal cancer, you can have that, but there's also MCED testing. Well, why is that important? Well, because you can't predict what cancer you might get, and you're at risk for all of these cancers, not just one. So that's what we're trying to get people to think about. And the second thing is how simple the diagnostic workups are. It's very easy to look at the cancer signal of origin and say, okay, I know what to do next now.
Yeah, Dr. Klein, you've done a remarkable job explaining and clarifying, that pathway and the role of MCED testing. Are you finding as the exposure of this test out in the community is increasing that message is equally well understood in the primary care community? Is it resonating? Are you starting to see uptake where you haven't done tremendous outreach and education?
Yeah, it's getting there. I mean, it's a slow process. But it is getting there, and there are an increasing number of providers who are ordering MCED tests and seeing their value. And there are a number of independent publications in the literature. I mentioned Mayo Clinic published their experience. Dana-Farber now, Mercy Health System, which is a big health system in the Southeast US, has published some of their experience. And there are more coming. And we have published, or we're about to publish in Nature Communications, our experience with the first 110,000 or so Galleri tests that were used on the open market. And again, as I mentioned previously, we see robust test performance in the real world across dozens, if not hundreds, of physicians who had used it. We see that they easily adapted to understanding the cancer signal origin prediction and doing the appropriate diagnostic workup. We've seen that the cancers that are being detected are largely ones that are not detected by standard of care screening, right? These are all things that we want for this test to be able to do in the general population.
I was just thinking about the reimbursement. Usually reimbursement is held up mostly by evidence of clinical utility.
Yes.
Right? And I know you guys have thought a lot about this. Can you tell us a little bit about, what your steps are towards showing that?
Yeah, so we're trying to broaden the definition of clinical utility as far as screening goes, because in the era of single cancer screening tests, clinical utility was defined by whether or not the test reduced mortality. But we think that that's far too narrow a definition. And we're trying to get all the stakeholders, patients, advocacy groups, the American Cancer Society, all the companies working on MCED are trying to have a unified voice on this. There's real advantage to being diagnosed at an earlier stage than a later stage. How do we know that? For virtually every cancer, survival rates are better for early stage, right? So survival for stage 1 is better than stage 2, survival stage 2. I mean, this is true. Second thing is the burden of therapy is lower if you're diagnosed at an early stage, even if you're not cured, but hopefully you are cured, right? It's less intensive therapy. It might be surgery only if it's really early stage. It might be surgery and some chemotherapy, but less chemotherapy for stage 2 than stage 3. You might be able to avoid radiation treatment if you are diagnosed at an early stage. If you have a head and neck cancer that's diagnosed at an early stage, the surgery that you get is far less mutilating than if you have an advanced stage cancer. So the burden of therapy is a lot less with early stage diagnosis, even if it has no effect on mortality. Third thing is there's clear economic evidence that shows that individuals who are diagnosed with early stage cancers lose less work time than those who are diagnosed with late stage cancers for a variety of reasons. Well, that's better for the patient. Obviously, they get to continue to work. They get to continue to have family time. They get to continue to be economically productive. Works better for the employers too, because work time lost is expensive. It's expensive to replace people and train them up, that sort of thing. In terms of the cost of treating early stage cancer versus late stage cancer, for most cancers, it's less costly to treat early stage rather than late stage cancers. Katrina Whitaker at the University of Surrey really said it very nicely. She said, people diagnosed earlier with cancer are not only more likely to survive, but more importantly, also to have better experiences of care, lower treatment morbidity, and improved quality of life compared to those diagnosed late. And so in the aggregate, clinical utility is more than just reducing mortality. Another potential clinical utility point is preventing a cancer diagnosis for the first time in the emergency room. So I've seen lots of patients in my career who presented with cancer for the very first time in the emergency room because they had a bowel obstruction, or they had a biliary obstruction, or had a renal obstruction. And they ended up needing a surgical procedure to relieve that obstruction. And then they ended up going to palliative care because there wasn't anything more that they could do for them. So if we can keep people out of the emergency room, and that's one of the endpoints that we're going to look at in some of our trials. When you add all that up, there's a lot in addition to reducing mortality. Now, let's talk about reducing mortality. It's not that I'm not saying that reducing mortality is not important. It is important. It is. The NHS Galleri trial is going to follow individuals for 6 years after screening, and at that time, roughly, is going to be able to model, not actually observe probably, but model what the expected impact on mortality would be. And hopefully, and I don't know this for sure, hopefully they'll be followed longer than that so you can look at actual mortality. And the other challenge in looking at mortality is it takes a long time for people to die of cancer when you screen them. And so the NCI, is doing the Vanguard study, and if they get funding for it, which isn't a certainty, in about 2 years is going to start a randomized trial that has mortality at the endpoint. Well, the problem with that is that's 2 more years down the road. Let's say it takes 10 years to get the output. So you're looking at the mid-2030s before we know, if you only use mortality as the endpoint, whether or not this stuff works. Well, here's the challenge with that. We lose 600,000 people a year due to cancer now. We've shown in multiple studies now that we can detect cancers that people die from that aren't covered by standard of care screening. Why don't they deserve the benefit of this? And the second thing is that the incidence of cancer worldwide is predicted to double in the next 15 years. So you want to wait. I mean, I find that status quo unacceptable because of those things. And then the last thing is that we hope that the technology will be iterated sufficiently so that by 2035, maybe the current technology we have is going to be completely outmoded and won't be relevant anymore, right? And if we get a mortality endpoint in 2035 says, yeah, this great, it works, but we don't use that. And that happened, actually. There was a screening trial for lung cancer using chest x-rays. And I don't know, it took 10 or 12 years, I don't remember exactly how long, maybe longer, to report out the results. Well, what happened in the meantime, low-dose CT came along. And so the screening modality was being tested for a mortality endpoint in this big clinical trial wasn't relevant anymore. So that's why we need to get the world to focus on the fact that, yes, mortality is important, but there are many, many, many other important things that MCED should be judged by. Another whatever new screening, it's not restricted to MCED. It will be for all new screening technologies.
In many disease states, we've been able to find surrogate endpoints.
Yes.
Right? Like in HIV.
Yes.
My experience was it took a while for people to be convinced that HIV viral load was a surrogate for
Yes.
AIDS.
Yes.
But once that was the case, all the clinical trials changed and management changed.
Exactly.
So, is there a morbidity surrogate for mortality here?
Yeah, that's a great question. That's really hard to define, but I've sort of hinted at it. Yes, it makes sense that there'll be less morbidity for early stage cancer. But here, there is a debate about this, a surrogate endpoint. And it's one that I mentioned previously, which is the reduction in late-stage cancer incidence in a screened population.
Mm-hmm.
And there is a huge academic debate about this. There have been 3 publications on this, high-quality publications, that have looked at 33 big screening trials, not using MCED, using other sorts of things.
Mm-hmm.
The first paper that was published from a group in Europe said reduction in late-stage cancer is not a good surrogate for mortality. Then another group that actually was led by someone at GRAIL looked at all the same trials that first paper looked at and came to the exact opposite conclusion. And the reason was that the first paper looked only at mortality when the trial was called and the primary endpoint was reported. And the second analysis looked at the long-term follow-up, which makes the most sense to me. If you want to know people are going to die, you got to follow them for a long time. So the groups have gotten together. The third paper looked at similar studies and so forth, and they have suggested that potentially reduction in late-stage disease is a good surrogate. They didn't come to that conclusion, but if you look at the regression plot, I think it's pretty convincing that late-stage reduction is going to be a good surrogate for mortality. And then the argument is going to be, well, it works well for some cancers, but not all cancers. And so there are some cancers that won't benefit from that, to which I go back to the point of that's fine. MCED should be evaluated in the aggregate. What happens at the population level for all cancers, not what happened in breast cancer or happened in lung cancer. So there is that debate about a good surrogate.
Right. Wow. This has been a great conversation.
Eric, we haven't met before, but you do a remarkable job of explaining very, very difficult to understand topics. I look forward to following you, and I hope you get out there much more broadly. And I'm presuming that you would like to do this as sort of a champion or an advocate in this space. I mean, you do a
Yes.
great job describing. Thank you.
Yes,
Yeah.
you're welcome, and thank you.
Thank you very much. This has been a very informative session, and I'm sure our audience will appreciate hearing it as well.
They will. They will indeed.
Halteres Presents is produced by Halteres Associates, a US-based bioscience consultancy that provides strategic and tactical services in the areas of diagnostics, medical devices, and life science research to clients of all sizes. The views, information, or opinions expressed during the episode are solely those of the individuals involved, and Halteres Associates is not responsible for any errors or omissions or for the results obtained from the use of this information. The information provided in this episode is for informational or educational purposes only and is not intended or implied to be a substitute for professional medical advice, diagnosis, or treatment. Halteres Associates would like to say thank you to this episode's guest or guests, and thank you for listening to this episode of Halteres Presents.